Hello, and welcome once again for another episode of Profiles in American Funded Research. I'm excited to have my guest today, Daniel Conway. Daniel got a bachelor's degree at Rice University, a PhD at the Georgia Institute of Technology, did a postdoc at the University of Virginia, became an assistant and eventually associate professor at the Virginia Commonwealth University, and now is a professor at Ohio State University. Did I get everything correct there?
Yeah, that's correct.
And I invited Daniel on today to discuss a project that this is the NIH project that he has met. He has multiple, but um, cell junction and nuclear forces as mediators of epithelial cell homeostasis is the title of the project. And I was wondering if you could just start us off with a brief description of the project, if you don't mind.
Sure. So I am funded by NIH is divided up into institutes, and I'm funded through General Medical Sciences. And so while I can't speak, maybe wonderfully about what their missions are, what they consider their missions are, from an applicant's perspective, I view them as funding really sort of fundamental basic science, and that's what I really built my lab around. I'm an engineer by training who has had a long-standing interest in cell biology, and I think one of the unique aspects I bring to that is thinking like an engineer and trying to build tools to answer questions and understand how things work at a cellular level. My group is one of the tools that we use are biosensors. And so these are protein-based things that we express in cells, and we can gain information about how the cells behave and work. And in particular, one of the things that I've been really fascinated about is how mechanical forces at the protein level are transmitted throughout the whole cell and how this could influence cellular behaviors. And for me, epithelial cells have been a real interest. Epithelial cells line, you can think about it as lining the skin and the lungs and these types of tissues. So really the insides and outsides of the body. And anytime you hear the word duct or gland or tubule, so you can think about things like the liver or the pancreas or the kidney, there are these epithelial cells that line those, so almost like the plumbing, internal plumbing in these structures. And these cells turn over really frequently, and how they get maintained in homeostasis is a really sort of important and interesting question. And so that's really what this project is about, trying to understand how cell-cell adhesion, so how they're coupled together, which is really fundamental to how these cells work, and maintain a barrier to allow things to move through and certain things not to move through. And then we're thinking about how those forces can go all the way down to the nucleus, where they may affect fundamental processes like gene expression or things moving in and out of the nucleus.
And I think that it's interesting that you came from an engineering background, and I think it's kind of, from my view, it's very important to have this kind of cross-pollination between fields because there needs to be a link between different in order to solve difficult problems between people with different strengths, I think. So I like that a lot. Um, could could you explain just for the general audience's purposes what homeostasis is?
Yeah, so at least the way I think about it is sort of keeping everything in equilibrium. And as an engineer, we talk a lot about this because, if you have a tank of water, right, and you have the water flowing in at the same rate as the water flowing out, right, the level of the water would remain constant. And so that when you think about something being in homeostasis, right, it doesn't mean that things aren't leaving or entering the system, right? It just means that it's sort of perfectly balanced. But you can imagine this is a real challenging problem, right? If you have a bathtub that's draining slow, how could you set the shower to be exactly the same rate, right, and not overflow the tub or or let the tub completely drain out? And so I think about that from a sort of a cellular problem. You can think about it just in terms of the number of cells, that's really important, right? To have, something like 50 to 70 billion cells a day turnover in the body, right? So if you're slightly off in terms of the cells that replenish it, right, you can end up with situations where you have too few cells, and that would obviously be terrible. And it's not that hard to end up in a situation where you have too many cells, right? And I think the obvious example of that would be cancer, but there's other diseases like psoriasis and things where cells proliferate too fast as well. So but it's more than just the number of cells. It's really about maintaining the cells in their correct orientation and architecture within the tissue, and then also the cell itself. How do you maintain its position relative to the other cells? How do you maintain its coupling, mechanical integration across the other cells? And so that to me is what really homeostasis means. And I guess I'm maybe a little bit different than some researchers. I know some people are really fascinated with a disease, or they want to study cancer, but sometimes I've had other people who think like me put it this way, it's fascinating we don't get more cancer. Which, there's a lot of sort of interesting things happening in the body at the cellular level and the tissue level that are helping to prevent things from going awry, right? And so those are the processes I'm really interested in trying to understand.
So you're interested in how the body kind of controls against these kinds of different like diseases and things from occurring, like producing too many cells or things like this.
Yeah, no, that I think so. And I think some of what we have started to do with some of our models, so we use a lot of, I guess we call it in vitro models, like where we can grow cells in a dish in the lab, right? And then we do a little bit of animal work also with mice. So it sort of blurs the line sometimes between homeostasis, but then we're also interested in sort of these developmental processes of how do you get this ductal structure and what happens and how do the cells establish things on the top surface versus the bottom surface or the apical and basal surfaces of the cell. Like how does this happen and how do the cells come together and meet and how do they organize into tissues? So, sort of intertwined in this question of homeostasis of how did we get to the equilibrium, right? And then when we shift out of the equilibrium, how do we return back?
Yeah, I think that those are very interesting questions. Um, since I think it probably will be uh, interesting to discuss, and it seems to be a part of this project, and from my own personal interest because of the physics background that I have. Um, could you discuss the Forster energy transfer, or it's also called fluorescence energy resonance energy transfer technique? I find that technique interesting.
Yeah, I mean, I think for me, I'm not a sort of a physicist, right? So I sort of use this technique as a way to measure distance between two fluorescent molecules, right? And it's sort of this relationship that when they get really close together, right, on the nanometer scale, the energy transfer happens more frequently. And so this allows us to this really goes into our biosensors, right? So sometimes I describe our biosensors as sort of miniature strain gauges. So we have two fluorescent molecules, and when this strain gauge is not loaded, they're really close together. So they're connected with an elastic peptide. And then when force is placed across that, the distance increase, right? This is sort of the basis for how like a bathroom scale would work, right? It's the spring. And so engineers and physicists have known for a long time, right? You can relate distance and force if you have something elastic. And fret isn't just about measuring forces, right? I mean, there are a lot of other biosensors where people have sort of dealt with the idea that if you want protein A and protein B in the cell, and if you want to see that they're really close together, you can use the technique. Or there's a lot of other conformational sensors that might be used to detect other things of interest in the cell, like calcium levels or different things. And so it's sort of this idea, or what I don't know the history of FRET, other than it was sort of a theoretical idea. And then as people develop fluorescent molecules for biological purposes, people were really excited about this because it really allows you resolution on the nanoscale, right? And it turns out that nanoscale is really important in a lot of sort of cellular or subcellular processes.
Yeah, I think it's very difficult, at least from what I understand, very difficult to get nanometer resolution. And there's been like a lot of focus on getting that kind of resolution on in a variety of things. So I think that it's definitely an important technique that kind of has allowed us to shed light on at that level of distance. And the fact that you can, I mean, you don't directly measure force, but you can back out what the force is from that technique is interesting. I think you already hinted at this to begin with, but maybe we could explore a little bit deeper. What are epithelial cells? Because whenever I think of epithelial cells, I think of the skin, but I know that after reading more about, from reading from this, it goes deeper than just the skin. And why are they important?
Yeah, I think I guess there's several ways I think about it sometimes as an engineer. I think one thing that's interesting to me is they are such a dynamic structure. And so if you, for example, take epithelial cells in a dish, if we grow them on an elastic substrate, if we stretch them, right, they start to proliferate, right? So that kind of was what drew me to them, right? Is they sort of have these dynamic processes. And my training was always in endothelial cells, which line the blood vessels. So I think there's a lot of sort of similarities between the two. Yeah, I think it's great that people think about skin, and I think people have sort of this intrinsic idea that, skin gets damaged, it has wound healing, it turns over. Of course, everyone knows that, the more sun exposure you get, right, to carcinogens and things can also cause cancer. So it's a pretty good proxy, I think, for things throughout the rest of the body. Um, but I think it probably doesn't take a leap of faith for people to sort of understand that there's probably a lot of parallels if you just think about the lung, for example, right? When you breathe in, right, you want a really strong barrier of cells against that outside air, which presumably has pathogens and other things that we don't want in the body, right? So that you want to have a system where you can sort of protect the rest of the body from that, right? So you can almost think about your lungs like your skin, right? We don't want things to get in. And then you could sort of extend that knowledge to maybe the stomach and the intestines, right? As food passes through as well, right? That you want sort of this really fundamental barrier function. But then, of course, there are all these other fascinating epithelial types, right? Like for example, these ductal structures, or you can think about the kidney that has these tubules, right? Where again, you want a separation between the organ and then this ductal structure, right? In the case of the kidney, where you're filtering things in and out of the urine, right? You don't want to filter it and then have it leak back. So I, as an engineer, right, a lot of times I see, okay, maybe the skin doesn't sound like a kidney epithelial cell, but from my perspective and sort of thinking about the fundamental properties of these cells, it's like there is a lot of similarity, right? And so oftentimes I find myself trying to figure out, well, what is the right model system, right? So if we study these basic processes, how do we, what is the right place to study them? And how do I, one of the challenges I guess I face as a researcher that does basic science is how do I connect it back to something more medical or more physiological that would maybe gain people's interest and appreciation in the types of things that I'm doing?
And what kinds of things can cause these epithelial cells to increase in permeability? Because I noticed that that was something of interest, I think. And I mean, obviously, I can see even just using the analogy of the skin, you don't want it to be too permeable, right? You don't want a bunch of disease easily entering your body. So
Yeah. Yeah, so I think if you start with the skin, I think there's a lot of actually pretty severe blistering diseases where there's mechanical defects in different proteins. And so you can envision, probably everyone's had the experience where they walk in some bad shoes and they get a blister or something like that, right? So everyone knows too much mechanical trauma on your skin is not a good thing. And the other thing I always, when I teach students, right, I tell them, you can pinch the skin on your arm or your hands and it can undergo a really large strain, maybe let's say 300% or something, three times in length. So skin is quite impressive, mechanically. But I guess I would say, bigger picture, if you think about what would happen if you lose barrier function, I think probably a lot of people maybe appreciate it better now that we've kind of gone through this pandemic with COVID. But even prior to that, there's a number of diseases where when you get a large infection or other inflammatory process in the body, sometimes too much inflammation can cause these massive sort of barrier losses across multiple organs, right? And typically the medical word we would use is sepsis. And oftentimes this would result in like a lower blood pressure and a pretty severe outcome, with really high incidence of mortality. And so typically the way that people maybe remember COVID, right, is a lot of people got put on ventilators, right? And so what happens, it turns out in the lungs, is when you lose this barrier function, not just in the epithelial cells, but there are endothelial cells or blood vessels, capillaries right below. When all of these cells become very leaky, then liquid moves from the blood into the lungs, right? And then what happens is just a little bit of liquid is enough to inhibits oxygen transfer from the air into the blood. And so it's interesting because there's not a lot of good approaches once this barrier is disrupted in order to fix it, right? I mean, of course, you can reduce the inflammation, but that doesn't necessarily get the cells to stick back together really easy. And so it's sort of a big problem. I think it was a big problem even before COVID that people that enter into some kind of acute lung injury or septic situation, once you go on a ventilator, it was always 50-50 whether the patient would make it or not. And I think people probably saw that during COVID as well, right? That once it got so severe, right? So that maybe gives people sort of a background about, why is this barrier function so important? As an engineer, it's like, well, what if you don't have it, right? Well, you end up with these situations where things don't, really fundamental processes don't work correctly in the body.
And outside of the skin, what like inside the body for interior organs and whatnot, what kind of examples are there of where you would experience mechanical tensions in the cells? Like what would cause
Yeah,
that?
yeah. So one of the projects that we had when I first started my group years ago back at Virginia Commonwealth University, we were really interested in sort of asking really basic fundamental questions. And so one of the things you can do with epithelial cells is you can grow them, as we call them 2D monolayers, but you can grow them as a flat sheet on a dish of cells. And this is really easy for us to grow them and propagate them that way in the lab. It's also really easy for us to image them because, again, they're in a nice 2D monolayer, and so it's much easier to image it. And as I was writing grants and talking to people, I got feedback a couple times of, this really isn't very physiological, that these ductal and glandular structures I mentioned are more three-dimensional. And so people can grow them in the lab in these 3D spherical structures, and the cells behave different. And so we thought, well, maybe this is an opportunity to take some of our biosensors and ask this question: what would be the force differences between cells grown in 2D and 3D? And it actually surprised us in the end. Maybe I didn't know enough of the biology, but it turned out when we put them in these 3D environments, in these very compliant matrices they grow in the sort of extracellular proteins that sort of support it. It's almost like it's a collagen gel, so it's a bit akin to like a Jello. They had much larger forces in this very soft material, and we thought that's crazy. I mean, how does that work? What's providing the resistance as the cells sort of contract? And it turns out that when they form these spheres, it's a bit like a soccer ball. So the cells are on the outside, and in contrast to a soccer ball, which would have air in the middle, these spheroids have a water-filled luminal space that we call. And so it's full of water and ions. And so this whole project, where we got this unexpected result that there were larger forces in 3D, really helped us understand the role of osmotic pressures in forming that lumen and driving that expansion. And then the osmotic pressure we think is really important in sort of regulating the stability of that duct. And so you can sort of envision if you have a bile duct or a tube in a kidney, you're going to want it to be a certain size. You're going to want it to have pressure to hold it open, right? But then obviously too much pressure could either tear the cells apart or in some cases may actually stimulate them to proliferate, right? So one good example of a disease where we think too much pressure would be bad is actually in this disease called polycystic kidney disease, where the kidney actually develops really large cysts and the kidney can grow to maybe 3 or 4 times the normal size it should be. And so we're really interested in those mutations in that disease, and do they help stabilize or sort of help set the set point, for sort of homeostasis of these forces. And then maybe the opposite disease of that we know is a defect in these osmotic gradients is cystic fibrosis. And so a lot of people have heard of cystic fibrosis as sort of a breathing problem, and that's true because these patients, the chloride, it's an osmotic agent, but this ion that's secreted outward, sort of into the lung airspace, it recruits water, right? And this helps maintain the mucus at sort of a set viscosity. And as clinicians have gotten better at treating this disease, right, these patients live longer, we're sort of realizing there are other defects in these patients as well. So sometimes they have liver problems, they can develop diabetes, and the men are actually completely infertile. So the ductal structures in the male reproductive system don't develop correctly. So I think there's a lot of examples of where you don't want too many forces, right? It's going to change the morphology, but not enough force is also really critical, right? So how do the cells optimize this and get to the correct set point? And you could probably envision that maybe different tissues or different organs may have different set points as well.
And if we go down to, I'm just curious if something like sickle cell anemia is due to an imbalance of force, or am I off base there?
Yeah, so I haven't done any research in sickle cell. Sometimes we talk about it in class because I teach an intro class to our biomedical engineering students, and so we talk about fluid flow through blood vessels, and then we sort of highlight sickle cell as sort of a defect in the shape of the cell. And so my sort of limited knowledge in this field, like I wouldn't say I'm an expert in it, is sort of that aberrant shape. When you get down to the capillary level, so the smallest blood vessels that we have, oftentimes they're only as wide as a single red blood cell. So if you have any sort of defect in that shape and it sort of gets jammed up, right, then my understanding is that jamming of the cells, whether they stick together with one another or they're too large to pass, creates some, I guess, back pressure. And, if you think of these capillaries as pipes, and I think that creates some of the situation with the pain for these patients, right? Maybe it's the pressure or maybe it's the lack of oxygen in the downstream tissue. Um, but I don't know. I mean, from my perspective, the red blood cells are pretty inert, right? They don't have a nucleus and they don't proliferate. But certainly they probably are mechanically responsive. I just a little bit beyond my scope of knowledge.
And could you explain, because I think this was in one of the papers you pointed me to, the what the nuclear pore
Sure.
complex is?
Yeah, so this is actually a new direction that we have gone in, and I think one of the challenges sometimes the biologists sometimes in the field joke, that I'm an engineer and I'm measuring things, but I don't know what it means, right? And so sometimes I'll tell people the story, if you're sitting in a chair, right, I could measure the force on each leg that you sit on, right? And in the end, you might say, but why is that important, right? Why did you build these contraptions to make these measurements? And so I've taken that to heart and really tried to think about when we're measuring mechanical forces on the outside of the nucleus, why would those be biologically important? And so one of the things, obviously, is the nucleus holds all the DNA, right? And every cell in the body has the same DNA, but yet all the cells express different proteins, right? So people, of course, are very interested in how the DNA is organized and how maybe mechanical forces could change that organization to maybe change gene expression. But more recently, people have gotten really interested in this nuclear pore complex, which is really the gateway by which how things move in and out of the nucleus. So the nucleus is really meant to be kind of a restricted space for the cell. In particular, when you think about a virus, sort of hijacking the cell, oftentimes the virus has to reach the nucleus, right, to really allow certain outcomes to happen with its sort of replication. And so the nuclear pore is going to play a really important role. Every gene that you need to make, you have the DNA, and then it's transcribed into mRNA, and then the mRNA has to leave the nucleus, right, to go out into the rest of the cell to make the protein. And so obviously all this mRNA has to leave, right? And then you have certain proteins that are made outside of the nucleus. They have to come in to accomplish their functions. And so the nuclear pore, I kind of think about the nucleus almost like a wiffle ball. I don't know if these analogies are helpful, but if you think about a wiffle ball, you have all these little holes. So if you wanted to move things in and out of the center of the wiffle ball, right, then you would, the bigger the hole, the easier it would be, right? But of course, if you didn't want a lot of things to move in and out, maybe you could limit the number of holes or maybe change their diameter so it's a little bit smaller. And so if you think about the nucleus as sort of made of an elastic lipid membrane, right, then you can think about maybe mechanical deformations of this structure could cause those holes to get bigger or smaller. Um, and this is not exactly what is sort of taught in sort of a cell biology field. Most of the ideas are that these holes or these nuclear pores are just there, and there's other proteins that are going to really be the gatekeepers to allow things to pass through. And so there are a couple people in the field, right? And I think increasing numbers of people who are starting to realize that the diameter of these pores is changing. And oftentimes the diameters of the pore is changing when we see mechanical forces changing in the cell, right? But of course, and then we sort of saw this as an opportunity. Well, we can build one of these biosensors in the nuclear pore, right? Then we can really start to understand, okay, is it force on the nuclear pore, right? And is this really going to affect how fast things move in and out of the nucleus? And I don't know if you'd asked me the next question, would that really matter? I don't know, right? I mean, we're doing kind of really basic things. We see certain things in the literature, for example, like cancer cells maybe will have more nuclear pores, right, in some situations. So would the rate at which things move in and out of the nucleus affect how fast the cells grow or some function? We just don't know. But these are things that we sort of think about in that area.
Yeah, so this is interesting because it seems like then potentially the cell could have a way of controlling how much is flowing in and out at different times. So maybe at a different point in the lifetime of the cell, it's important to have a bigger hole or something like that. So it seems like it could be very dynamic in that way. Or is it just
I think
static
so.
as
I
far
mean,
as you
certainly
know?
there's a lot of nuclear pores, right? We don't really know. I mean, we have a cell line where we have been sort of mechanically perturbing the cell and trying to understand under which situations does the force go up, under which situations does the force go down? Are those things correlated to how fast things move in and out? Just maybe for some reference, right? I think there's, ballpark like thousands of nuclear pores, right? So it's not the case where you just have one hole. But certainly there are sort of interesting questions about are all those pores active? Are certain sides of the cell more active than another? You could think about a cell moving. One question I have, but I don't have the techniques yet to answer it, is are some of the pores focused in one direction versus another? So I mentioned that things move in and out. And so again, like I'm an engineer and I'm always looking at things that I experience in the real world and would they apply. So if you go to a football game, for example, they might have a dedicated entry door and an exit door, right? And that's going to help with the crowd moving, right? If we all have to move through the same door, we're all going to have to take turns and wait, and so it's slower. But I don't know, right? I don't know if that's really what's happening at the level of the nucleus or not.
And how would you go about measuring uh, that? So how could you tell if there was maybe more doors on one side or another? The flux, how would you get measurements of
Yeah,
that?
so I think yeah, so that I mean this was sort of the idea I pitched for my NSF grant, right? Which is to really try to build some biosensors. And the ideas that we have would be to create something in the middle of the pore that only binds something when something passes through, right? So it's sort of like, again, I guess if we use the football analogy when you walk through the door, right? If we were to spray you with some paint or something like that, then later on I could say, okay, you walk through that door because you have that paint. Or if you got stuck in the door, right? We trapped you, right? So we're sort of looking for some kind of tool where we can directly image, right, the entire cell and ask this question, okay, did all the pores get labeled? Did a fraction of them get labeled? And these things, on paper are really easy. It's nice to draw a cartoon, but it's really hard, right? I mean, I think that's sort of the challenge I have with biology is it's so small, and we build things, right? But then one of the challenges, even for me, is as we started this project, the nuclear pores themselves are really small. So even as we develop tools to do this, we sort of realized that our microscope wasn't good enough to resolve the individual pores, right? And so then we have to start talking to other groups who have the tools to be able to measure something that small and get the resolution where we can distinguish one pore from another pore that might be right next to it, right? And so it's frustrating at times, right, to have the idea, right, but then to realize that it's not so simple.
Yeah, I think also that actually I know that this isn't the science necessarily directly, but that highlights is the importance of collaboration, I think, because you don't necessarily have all the tools in your lab available to answer all of these questions, so you have to sometimes collaborate with others to do that. And I think that's a really important part of science.
No, I agree, and I think I've been really lucky. I oftentimes think about the people that have worked in my group, the students, right, and the other trainees that I've had and the staff, of getting the right people that are willing to do things. But then sometimes these collaborations are oftentimes really random. You meet someone and I have 5 or 6 projects and I don't want to discuss them all because I sound like maybe I'm not well focused. And how do you figure out, right? Do they have a similar interest? Do they have a technique that could be used? Or are they a nice person and willing to work with me and be patient on something that maybe isn't quite fully developed. And so there's sometimes seems a lot of randomness, right? And I think putting oneself out there and knowing when is the right time to talk about an idea, because you always worry that maybe someone would steal your idea, right? Scientists are very excited to get some credit, right, when we do something. But then at the same time, just making sure, when is the right time to present it, right? Because if it's just an idea, ideas are really cheap, right? But then you really want to be at the stage where you actually have some data, and then if you propose this collaboration, right, you're really capable of doing it. I think oftentimes I don't always particularly like the process of traveling and going to meetings, so I sort of enjoyed COVID when I could stay at home. But then I really realized that, yeah, some of these opportunities oftentimes come from going to a meeting and talking about data and meeting new people. And, I think that's a really important process of science.
Yeah, I've always felt that that was the actual only reason for conferences in reality is eventually you would like to form collaborations. I mean, maybe a small piece is, telling other people, hey, this is a cool work that I did and try to get citations or something. But I think the big piece is really, finding collaborators. Um,
Yeah, I certainly think you can't do it by yourself. And I've tried to like lower my, I don't know what it's like, fear of contacting people I don't know. And so oftentimes when we get strange data and there's an obvious person, a big person in the field. And it's like, well, I guess I'll email them. And sometimes, but do you know them? No, I don't know them. But I mean, I joke. It's they're sort of like phone a friend, right? But sometimes when we have something and we have a problem, it's like, okay, well, who could we contact for help? And everyone's busy, and sometimes they don't answer your email. But I've honestly had really great luck, even if it doesn't lead to a collaboration. I'm just saying, hey, we have this idea, we have this. What do you think about it? And I think most scientists are really excited about their research, and I feel that way whenever someone reaches out. I mean, even when you reached out about doing this interview, I thought, well, this is really cool. Someone noticed who I am and is interested in what I'm doing.
Now, um, just steering quickly back to the science that you were doing. Now, when I think about the cell and even bigger than the cell potentially in the body, I typically think of there's some kind of, let's say, Brownian motion, randomness that's associated with this. And so how does that affect
Sure.
your ability to peel out, and like what do you have to use like statistical techniques to kind of help you decide if something is real or not that you're seeing?
Um, yeah, I would say it sort of depends on the level. I think, in terms of Brownian motion, I'm thinking about individual molecules diffusing, right? And so on a larger scale, we don't think about cells, for example, jiggling around too much, right? Particularly if the cell is mechanically integrated to the surrounding cells or the surrounding protein networks in the cells. So, but certainly I think about noise in general, whether it's Brownian motion or it's maybe just the resolution of our microscopy or our other approaches. I would say it's frustrating. I don't know if this is where you wanted to go in this direction, but we do spend a lot of time thinking about reproducibility, and there oftentimes is sort of this underlying variance, whether it's day to day or cell to cell. Um, when we think about measuring mechanical forces, oftentimes we see two cells, and one has a high force and one has a low force. And I'm not able to articulate why there is a difference. We try to do different things like make sure they're each in the same stage of the cell cycle or they have the same surface area or things like that. And even when we control for the variables we think are important, we often find that there's sort of this heterogeneity. And what does that mean? Is that important? I mean, it's frustrating as a scientist. I don't want heterogeneity. I want precision. But I don't know, right? So certainly we have lots of times where we measure something and then the next time it's different and the next time it's different. And then trying to think about what does that mean? Do we just abandon it? It's difficult.
Yeah, I think sometimes those kinds of things that are really hard or seem impossible to explain are actually hinting at something deeper. And sometimes they're not hinting at anything at all. It's just I see that as a big challenge for experimental science, although I'm not an experimentalist, so I'm just doing it from my perch over here as a more theoretical person. But now, um, a major reason for doing this podcast is to discuss about, with the public also, where is the money going? Can you give an idea about you know, supply, is it going to supplies, people, travel, these kinds of things? What kinds of things is the money being spent on?
Yeah, so I think when I put down a budget, right, for a grant, oftentimes the biggest expense is the labor, right, the personnel. I think if we think about, I don't know how much you've gotten into this in other episodes, right, but a lot of research labs are funded by what we would call student trainees, so graduate students or maybe even postdoctoral fellows. And so these students, in sort of exchange for working on this project that ultimately becomes part of their thesis, the grant is used to pay them a small stipend and offset their tuition charges. And so depending on the university you're at, right, this can be, with the stipend and tuition, right, $40 to $50,000 kind of ballpark range. And so again, what does that mean, right? Well, I guess that's that full-time person, right, that can lead that aspect of the project. And in essence, as the PI becomes sort of this project manager, right, of managing the grant funds and managing the direction of the project and having, daily to weekly meetings with all of the staff and trying to, which is frustrating, right? Because I'd like to be more involved in the hands-on things. But then the supplies, yeah, I mean, certainly we use a lot of reagents when you do experimental biological types of experiments, right? So we have to buy a lot of reagents just to be able to handle the cells in a sterile way, to provide them with media or sort of growth nutrients for the experiments. Those are my big two line items. I mean, certainly we do go to a meeting or two, right? And certainly I weigh is it worth it to go to the meeting? It seems like, is that a necessary cost or not? And I also struggle with that as the mentor because I need my trainees to be able to go to the meeting and practice their presentation skills and their ability to communicate and interact with collaborators, right? So I can't just go to the meeting myself and sort of present all their work, right? Because then I'm not supporting their growth as a career, right? So I think some of the things that we do in these projects, right, for research are really also supposed to be training sort of the next generation of scientists, whether they go into academics or they go into industry. Um, yeah, but I would say for me that it's really the labor and then sort of these like consumable supplies.
And I haven't had a ton of people with NIH grants on, and I know that you've had both NIH and NSF grants. Would you be able to just touch
Yeah.
briefly on how they differ, how how the administration differs of those grants and such?
Yeah, so I mean, I can speak to my experiences, right? I don't want to speak on behalf of NIH or NSF. Um, you know, NSF is really focused on basic science, right? And so, even the basic NIH grants I write, I'm always framing it in the context of a disease or a problem, to sort of highlight that, right? What is the significance to medical sort of types of things, right? Even if you wouldn't call human development or homeostasis medical, right? It still fits in that human physiology. Whereas I think NSF is much more excited to take sort of basic questions, right? So that nuclear pore idea I had, well, can we measure the rates of things moving in and out, right? I don't have to frame it in a sort of a medical need to answer that question. This is sort of a fundamental concept that plants move things in and out of the nucleus, like lower level organisms do this, right? Animals and then all the way up to mammals, right? And so this is a fundamental question. How do things move in and out of the nucleus, right? And would mechanical force like increase or decrease the speed? Is that a new way it could be regulated? And so they're very open to that. And I think when I write grants or when I think about these questions, I have to think about what are the priorities of these agencies, right? And what would they do? So NSF is really nice, at least for my type of research, because they have a mechanobiology call out, right? And so I can take an idea like this where mechanics, regulating biological function, and send it to there, and they have a system where they have reviewers and program officials who are interested in that type of research. So I guess maybe that's one similarity between the two, right? Is no matter where you write grants, and I'm sure this is true even if you're not doing biology, you have to find an organization, whether that's a government organization or a private foundation, who understands the type of research you're doing and is excited and willing to fund that type of research. And certainly I've had other ideas where I've sent them other places or I have not found a place for them, right? But sometimes I send them places and it's not well received. And so ultimately, at the end of the day, because the research requires funding to do it, then those ideas don't go anywhere. And so I think it's not my position to say we should fund more of X or Y type of research, but ultimately, whether something is funded or not really does dictate what types of research occur.
Thank you for that. And um, what I always like to leave off with is a question that's sort of dear to my heart, which is, how did you get started on this path in science? If you could rewind back to, high school or earlier, uh, what 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 really got you started down the path of being a scientist?
Yeah, so both of my parents are chemists by training. So I guess I was raised by scientists. They never went on to grad school, so they worked in industry. But they always, it's sort of like whatever your parents did, right? You can't be terrible at that, right? And so definitely I felt like that as a young child that they expected me to do well in science, right? I couldn't bring home bad grades in science. And so I don't know if I get some of the curiosity from them about that, or if I was just sort of forced to appreciate science. I didn't do particularly well in like organic chemistry, for example, in college. And my parents had no sympathy. They said it should just be intuitive, right? And whatever that wasn't intuitive to me. I think my parents also maybe realized I was a bit more applied, and so they had sort of pushed this idea that maybe I should consider engineering rather than the pure science. So I'm really fascinated with how things work. And so just understanding how something happens, if I don't see the reason why to understand that, I think it's challenging for me to grasp it. When I started as an undergrad, I thought I was going to do chemical engineering because my parents were chemists, and then they said you should be an engineer. And I sat in these classes at Rice in Houston, Texas, and they talked about their graduates, like, I don't know what it was, 70% or something went to work for the oil industry. And where do you start in the oil industry? You start on an offshore oil platform. And I was just sitting there for about 2 weeks and realizing this wasn't exciting to me. And I don't know other people, but it just wasn't what I wanted to do. And I sort of took this leap of faith. They had this new area called bioengineering or biomedical engineering. And so I thought, why don't I try that? I've always been interested in biology. I've been interested in how the body works. I toyed with the idea of maybe going to med school at one point, but I just really liked what I was doing. And I ended up in grad school. I think my parents maybe had lamented they hadn't gone to grad school, and so they were supportive of that at the time. And then when I graduated, the economy was in a mess. I think it was like 2009. But I had always thought the dream for me would be to work at a startup. I really like this idea of doing new things, and um, it just wasn't the right time. So that's how I ended up in a postdoc, just kind of because of the economy. And I've really enjoyed the science. I really enjoy working with students. So I teach classes as well. And I don't know. So I guess I've lucked into sort of a career path that fits well for me. And I try not to think about the what ifs, if I had done something slightly different. I hope I would have been equally as happy doing something a little bit different.
Well, thank you very much for joining me, and I thought this was a great episode.
Well, thank you so much. I really appreciate the opportunity. It's always fun to chat about the research, and I hope I did a good job explaining it at levels that everyone can appreciate.
I think it was great.